just to sort of get this all in 10 minutes, I think we're just going to do a brief overview of the company and then sort of focus in on what we might be able to do with this technology from a trial design perspective. So just a couple of words on Locus. So Locus is the worldwide exclusive owner of CRISPR-Cas3 technology. And I'm sure many people know CRISPR-Cas9 from all the news that's been recently available on that. But instead of being a pair of molecular scissors for gene editing, the Cas3 enzyme works more like a Pac-Man. It essentially makes a small nick in one strand of the DNA in the genome of a bacteria cell, and it unzips, unwinds, and permanently destroys that single strand around the genome. What we do is essentially, through synthetic biology, we use a viral vector like most DNA editing technologies, and so we use bacteriophage. We open up the genomes of the bacteriophage and embed either CRISPR-Cas3 arrays, RNA guide arrays, which would activate an endogenous Cas3 system inside of a bacterial cell, or we'll take the full cascade Cas3 complex and load it into the phage genome for delivery into the human body. And we have actually advanced through a Type B meeting for a lead asset. I'm going to talk a little bit more about that. But that phage safety profile, coupled with the efficacy that we see with CRISPR-Cas3, we think is one of the big opportunities with advancing this platform for all kinds of indications, not just in infectious disease, but also in the microbiome. The company is focusing on 3 assets. I'm not going to go into great detail about each of the individual indications, more a focus on the different routes of administration. And so our first asset, we are going to be approaching trying to go after complicated urinary tract infections. The leading bacteria that we're after is E. coli. I'll talk a little bit more about that, but we're going after a route of administration that's intrabladder. We'll follow that up with a Pseudomonas indication, likely anchoring on a HAP-VAP lead indication through a nebulized inhalation route of administration, and then we'll follow that up with a Clostridium difficile indication for recurrent C. diff going through a gut route of administration. The idea here is to systematically build 3 different routes of administration to get into 3 zones of the body where most of the infections take place. We think there's some huge opportunity in being able to stack those routes of administration to be able to use that as we build the safety data sets against the platform over time. The company's done very well over the past 24 months. We've raised $21 million. Very happy to say that we have the funds in the bank to be able to advance our first asset through a phase 1b trial. So we will hopefully get to have that data set in hand of what that human PK is going to look like with these drug products. We'll also have money in hand to get a Pseudomonas indication all the way through an IND, and we'll be working in some of the CMC scale-up opportunities for the C. diff asset. It has some uniqueness to it as an anaerobic target. So to talk about trial design, well, I thought it would be really important to give a little bit more under the cover view into what a product is off this platform. And so our discovery process is very bioinformatics heavy. And so we will download hundreds, if not thousands, of genomes. We'll create huge isolate panels of clinically relevant isolates for the target bacteria that we're after. And what we do essentially is we search for conserved genes. We build RNA guides that can be arrayed so that we can actually build CRISPR-Cas3 command sets that can then be loaded in to trigger after targeting those conserved genes. Take E. coli, for example. We make sure we can cover all 180 serotypes so that when we build these RNA guides, they actually go after genes that we know that are throughout the population of bacteria that we're trying to kill. And then we also search for the corresponding phages that would essentially provide the right host range to those isolates so that we then can move into construction of the drug product, which is through synthetic biology, to open up the genomes of these bacteriophages and load in these CRISPR-Cas3 array commands. And we can essentially then take all of those drug product candidates through a battery of pretty standard in vivo tests: tolerance, efficacy, distribution, and persistence. And we have advanced through 7-day toxicology despite some of the FDA's flexibility in what you might be able to not do there. I'm not going to go through the data in grave detail, but I think the punchline is we very typically see 4 to 5 log killing above and beyond what you would normally see from the phage lytic cycle alone. And so we typically move something forward as a drug product when we get into the 8, 9, 10 log killing range to be able to make sure we can fully eliminate a bacterial infection in many times less than 30 minutes. So that's what a drug product from our platform would look like. I think it might be important to then sort of have that grounding to talk about the trial structure. So we're going to advance our first asset as a monotherapy into a phase 1b trial. We're going to do that. We're going to go after E. coli-based presence in the bladder. We're going to be targeting colonized patients, so patients that have not yet advanced to the infected state. And what we'll be primarily looking for is a safety endpoint, but also to be able to have a secondary endpoint of PK in human. And that should give us the efficacy indication. It's not obviously statistically powered with only having 30 patients in this initial phase 1b study, but that indication of efficacy is something that we believe we can then lean on for every other trial and every other asset that comes off of this platform. A lot to work out there with the FDA, but it's a decent set of assumptions in this serious and unmet need arena. When we move from that phase 1 monotherapy, it is very likely that it will move to a pretty traditional non-inferiority model for what would be in a phase 2 study. A concomitant dose with some form of standard of care, head-to-head against standard of care. But what we do think we can do is to look at a single species, multiple site approach. And I'll talk about that next, but what that would lead us to be able to get to is to potentially have assignment into the Tier C framework that would either reduce or eliminate the need for huge pivotal trials to get the drugs approved. And so just a little bit more on this theory, which I think this is where the Duke discussions have gone and where we really think we're able to leverage quite a bit of opportunity here. So we talked about the E. coli first step as a monotherapy to establish that safety profile. But other assets that then lead off of that from this platform, we believe we can have an anchor indication, let's say Pseudomonas hap-vap, but we can bring in patients that are cultured for MDR strains in other regions of the body, be it the bladder, be it the sinus cavity, be it the skin, so that we can get that patient population set up to a level that's potentially high enough that we could potentially have composite POC and small pivotal study groups together in the same trial. Again, a lot to work out there with the FDA, but we believe this is where the opportunity with phage safety really gives a huge step forward into the flexibility of how we might be able to design trials to essentially attack the escape pathogens systematically. And maybe a better way to really kind of understand this is to take a look at it for sort of a more traditional view of a drug development process. Let's say 10 years, $1 billion to be able to develop a drug. And at the end of that time period, you're essentially in a position where you have, for one zone in the body, the ability to eradicate a number of pathogens in that zone. Let's say complicated urinary tract infection. What we're going to be working on is to look at stacking a set of POC pivotal combined Tier C framework products that are bacteria focused across multiple sites of infection. And you get to the same endpoint to be able to have a platform of drug products that attack MDR-based escape pathogens, But you get that in a way where you're going at this in a far shorter period of time for far less cost, which inherently provides a lot less risk. And so with the safety profile of phage and the efficacy profile of CRISPR-Cas, we believe that we have a solution to be able to attack the escape pathogens in a way that no one else can. Again, a lot to work out with the FDA, but I think that was really the point and purpose of what we wanted to try to share today in the context of this discussion with the folks from Duke. We have probably all of the same challenges that Greg has, maybe even a few more. I think, we just talked about the trial design one. I think just a note on resistance, because it's always the first question that comes up when we talk about phage. We're really fortunate to have been assigned to the vaccines division of CBER. The thought process there about how essentially drug products can move through in more of an annualized basis to address the mutations that you're seeing out there with your drug product, that's the thought process that we think is needed to be able to figure out how to have a phage-based platform actually work for the long haul for society. And so while we don't believe that our drug products will be immediately resistant by any stretch, because they're cocktails of phages, we could easily see resistance within 2, 5, 10 years, and we'll need to pretty rapidly be able to address that. For us, we almost look at the CRISPR-Cas array as a drug substance and the phage as an excipient, or something that can be thought of along those lines, such that the phage delivery vector can essentially be rebuilt time and time again to carry those arrays that attack the conserved genes repeatedly. Manufacturing, I think Greg stated it well, and Greg's probably one of the most advanced in this space with his CGMP environment. Pretty impressive stuff, but we completely agree that this challenge does become a great opportunity. It takes a lot to put a manufacturing site in place. I actually, in a past life, was the Chief Technology Officer of Patheon Pharmaceuticals. So building a CGMP manufacturing facility for phage would become very much a barrier to entry to anybody else that could come into the market. And we think the last one is capital. It's been a tough space. Frankly, raising money in infectious disease alone is incredibly hard. Trying to do it with phage is even harder. But what's really been the anchor for us, and things have changed dramatically in the last 12 months, has been the acknowledgement inside of the strategic big pharmas that this platform has applicability between both infectious disease and the microbiome. And when we look at the safety profile of phage, the efficacy profile of CRISPR, and the ability to take that platform across indications that are both infectious disease and microbiome, we have a lot of interest from that group. And I think everybody in the room would agree that one of the big things we have as an opportunity as sort of a cluster is to entice big pharma back into this game with the checkbooks that they have. And we certainly hope to be on the forefront of being able to break that through. So thank you very much for the time. I know that was really quick. There's a lot of other information related to the company. But again, it's a pleasure to be here and look forward to the panel discussion.